Thank you very much. Thank you very much for the invitation. So the plan for this talk is that I understand that you already have talked a lot about place and grid cells, so I won't repeat much of that. I will have a few introductory reminder slides. Then I want to spend some time on models, or especially attractor models, for how grid cells and actually also other types of spatially modulated cells might arise. Partly related to what you already have discussed, especially in relation to head direction cells or directional cells in the fly, but mostly also because that may tell us a lot about neural networks in general, and it may be food for discussion, although much is still unresolved. Then I want to go over to more unpublished or very recent material, and especially spend some time on a new type of cell which is related to the firing to the position of individual objects in the environment. And then I will see how far I get. But before digging into this, then I will start out with a movie. I'll show that movie just as a warm-up, and this is a movie I usually present for completely different audiences, but I still like it, so let's show it. And then after that, when I begin, don't hesitate to ask. All kinds of questions are welcome, so and I'll just try to modify my talk so that I get through the most important things anyway. All right, so let's begin with the movie. That's it. So where is the system? What does it consist of? Well, you have heard already from Nahum a lot about place cells. You heard about grid cells. You heard that they are in different parts of the mammalian brain, in the hippocampus respectively, and the medial entorhinal cortex. You have heard that they are connected. Oh, sorry, I forgot the light. Yeah. So I don't want to go further into that. I do want to show a slide of a grid cell. I understand that the same cell was already shown by Nahum, so you then both saw how nicely regular it is, and if you look closely into it, you also see that it's not so regular, so we'll come back to that. The triangular structure is clear, and that also explains why we call them grid cells at that time. Work has shown that you can decode position pretty precisely from a composition of grid cells, especially if they vary in scale, for example. So that this is much of the basis for suggesting that these have contribute a metric to the brain's map, spatial map. So you also know, probably from what Nahum told, that they vary in phase. Phase means the XY position of the grid pattern, like the blue and the green one here. They vary in scale. So you see examples of that, two different scales in the green and the blue. And in the orientation. So, and actually there are further variations, like the amplitude of the different points, but I won't go into that. To some extent, phase, scale, and orientation are actually organized anatomically, but in different ways. So when it comes to the phase, you probably heard that phase is pretty, to a large extent at least, non-topographic. That means that cells that are anatomically close, like for example the blue, green, and red cell, which are picked up by the electrodes or the tetrodes above, still may have almost random relationships with regard to their phase, so that you see the green grid cell, and you see a blue one, and you see a red one, and there is no, at least no macroscopic organization of the scale of these cells. That may be different when you go down to the really close connections, But at a larger scale, over 50 microns, there seems to be no relationship between the XY position of different grid cells. When it comes to the scale, then you also heard that there's a dorsal ventral topography. So what you see to the left is a sagittal section of a rat brain. Red is medial entorhinal cortex. You start at the top, dorsally, and you have almost exclusively small-scale grid cells. That means that the distance between the grid peaks is small, and also the peaks themselves are small. Then, and these maps, I guess it's obvious based on what you saw yesterday that this is an overview picture of a box where a rat is walking, and each dot is a location where the cell fires, and then you see the grid pattern here. You see that the scale is small here, and then the scale increases as you go down on average. And you probably also heard that that it is organized in discrete modules, so that this is not a continuous change from small to large, but as you go down, you actually go in jumps, so that you have you can divide the grid cells into at least 4, probably a few more clusters of different scales each that are largely non-overlapping. So I'll come back to that quite soon, but just to finish the introduction, also mention head direction cells, which I'm sure you also heard a lot about. So discovered by Jim Rank first. These cells exist also in medial entorhinal cortex, so here. They do not fire at particular positions, but this is a polar plot showing firing rate as a function of the orientation of the head. So this cell fires only when the rat's head points to the left here. This one fires only when the rat's head points to the upper left. So those directional cells exist also in medial entorhinal. They are more abundant in the in the layer 3 and down, not so much in, or almost not actually in the rat in layer 2, where most of the grid cells, the prototypical grid cells are. In addition, there are border cells. You also heard about those that fire specifically along borders of the local environment. So again, this is a box seen from above. Firing rate is color-coded here, and you see that this particular cell fires only on the right side here, regardless of how much you stretch the box either this way or that way. And in a different environment, chooses the left side here, and you can even add a wall, and it fires on the corresponding side to that wall too. But always only along the walls, almost not seen any cell that actually has a field that is displaced from the wall. Finally, there are 2 more. So speed cells are cells that are mostly interneurons, but not exclusively. They have a firing rate that's shown here to correspond very much to the speed of the animal. So this is a rat running over 2 minutes, 120 seconds. Gray is the speed of the animal, and then color is for 7 different cells. Firing rate of each of them, and you can see that the firing rate follows very closely the speed of the animal. Maybe the most clear actually here for the yellow one is almost totally matches the speed profile. So and those cells don't really have any strong spatial modulation, but they fire, they have almost, at least within a large range, a linear relationship between speed and firing rate. And then finally, I should also mention that in this, still within medial entorhinal cortex, there are also many cells that are spatially modulated that don't have any very striking grid pattern, and just show 7 examples. Here you see that they are quite stable over time, so these are pairs of recordings, 2 different recordings, but of course one can assume that some of them might be grid cells that have so wide grids that you can't actually see the grid pattern, but that doesn't quite make sense in all cases. So even in large environments you don't see that. So there is probably a pretty large proportion of cells that are spatial but not grid cells. And again, they exist in medial entorhinal cortex in all layers, including layer 2 and 3. So perhaps what's most striking about the medial entorhinal cortex is that there are so many types of cells, because the brain doesn't really normally consist of types of cells. In this case, there is also some overlap. So there are cells that, for example, combine direction and space. So, for example, head direction and grid cells are expressed conjunctively in many cells. 1 second. Yeah. But still, it is pretty unique in the medial entorhinal cortex that one parameter so strongly predominates firing. This is usually, it's very different from the mixed selectivity, as we call it, or most other brain areas. Yeah.
Just to clarify, there are 4 of these cells that I am not from the field at all, but when I look at them, I think, okay, that's how place cells looks like.
Yeah.
Why are they not place cells?
Well, that depends on the definition of a place cell. If you call a place cell simply a cell that fires at one place, then they are place cells. But then hippocampal place cells are different in many other ways too. And I will show later, and I'm sure you saw yesterday, that one at least equally striking difference between place cells and all entorhinal cells is that the place cells remap. So they exist in all kinds of combinations. And I think that's actually the much more important part of place cells. Yeah, but I'll come back to it. All right, and then species. You've seen this many times, even on the movie. But this is then phylogenetic tree for mammals. The point I always like to make is that rats and mice are on a different branch than the bats, which is not always clear to people who work only with rats and mice. So they always think that either small rats or small mice. But the fact that they have not only exist, but they have so similar organization, brain organization, properties, and still are on different branches, must suggest that much of this has arisen quite early on in mammalian evolution, at least. So, and then maybe not so surprisingly, cells with similar properties have been found also in primates. They're not quite the same, and they don't look so nice, but there are many. It's easy to explain. For example, in humans, I mean, the quality of recordings is not the same. The experimental conditions, these are epileptic patients sitting in a bed and a kind of virtual environment, but they also see the real environment. So, but still, statistically, this pattern comes through. So I believe there's some truth to it. Yep, that's the introduction. Then let's move on to the mechanisms. And of course, now we walk into uncertain territory because we don't really know the mechanisms, but I think it's very useful to think about it. And you did that to some extent already yesterday, I heard, and also actually during 2 talks, not only Nachum's talk, but also Vivek's talk, because I'll come to that very, very soon. But as you heard yesterday, then it has been suggested for quite a while that you can get localized activity, localized in any kind of dimension, by arranging cells in a network on a ring. So it's called a ring attractor. And that idea has been around for quite a while, as far as I know, suggested already in 77 by Amari. But it was picked up in the 90s by multiple groups who all saw that this could actually explain properties of head direction cells. So 3 groups more or less at exactly the same time, Skaggs and McNaughton, and Zhang and Reddish and Turetsky, suggested essentially the same idea, namely that you can account for properties of head direction cells by arranging the cells conceptually on a ring so that cells that have a similar directional preference are next to each other and then are connected to each other with both excitatory connections, as shown here in red, but also, not shown here, inhibitory connections that go a little bit wider, so that and then what you then add is input, which is informs about the angular speed. So this is in the separate layer here. And then if there are connections to the head direction cell ring here, then those inputs can actually move the activity bump that's caused by these connections here around in accordance with the angular speed of the animal's head, either this way or that way. So you heard that yesterday, but this idea then, let me first say that for perhaps the best evidence, for certainly the most direct evidence for such a ring attractor is the fly work from the central complex that you heard about yesterday from Vivek. So such similar direct evidence doesn't exist in mammals, so one has to look for predictions that you would make with such models, and one is that with so strong connections you would expect that there would be a similar kind of coherence between which cells are active from one environment to the other, in the sense that two cells that have if one cell rotates about 180 degrees here, or this is 90 degrees from this environment to that environment, then you would expect a similar turn for the next cell and also for the next cell, so that they simply move as a coherent cluster. And that's exactly what you see in rats when they go from one environment to the other. So it's just, I mean, we often call this low dimensional, because it really is no remapping. The system keeps the same intrinsic relationships from one environment to the other. So that's where we are with head direction cells. But if you now go to place and later grid, so then these models came in the mid-1990s, and it was then suggested very, very early, soon after, by several groups, mostly McNaughton, that a similar arrangement could actually also explain properties of place cells. So it just has to be two-dimensional. So instead of one ring, which is one-dimensional, it goes in two directions instead. So what this is illustrated conceptually here. So that what you have here is each dot is one cell again, but they are arranged now in according to where in the spatial environment, in the outside environment, they are firing. So that a cell that two cells that are near each other, up here are the two cells that are place fields in this part of the box. Then again, just like in the ring attractor up here, then there are strong connections between cells with similar firing properties, similar firing places. So they can also be illustrated by this. This is a connectivity illustration, so strong connections between cells that fire in same places. And around it, again not shown, you also have a wider ring of inhibition, shown here, which then prevents this bump of activity that arises because there are so strong connections. It prevents it from just spreading out and completely going epileptic and firing everywhere. So if there's a ring of inhibition here, then you can keep this concentrated activity concentrated. It won't just go everywhere. But, and that's why it's called a Mexican hat, because it looks like a Mexican hat. It's a strong excitation locally and then a wider ring of inhibition. But then the point is, again, just like in the ring attractor, that if you have an input, and this time about the speed or direction of the animal, so the animal is moving in the real environment like this, then you can actually use that input onto this bump of activity and move it around in the environment in accordance with the animal's movement in the so that the network, the movement in the network becomes corresponds to the movement in the outer world. So this was the idea, still talking only about place cells, because at that time only place cells existed. But then one little modification, and when we get grid cells. So there are several ways to do the trick to get grid cells in such a network, but perhaps the most obvious one is to have in such a network, so again, each dot here is one cell. They are arranged. So this is a network. You can imagine it is seen on the brain surface, but they are arranged in such a way that two places that are local or close to each other, or two cells that fire in similar places, are put next to each other. So now you can imagine that in such a network, then you may have several bumps of activity. So that's illustrated here. And again, short range excitation in green and longer range inhibition. And if those arise spontaneously, what will happen? Well, that's essentially Turing's principle. Then they will organize in a particular pattern, or there are not many patterns that are stable if you have such competing bumps of activity. So it will organize in a hexagonal pattern, like you have here. And that's because they all excite each other, but they're all 2 seconds, yeah? And then they inhibit each other, and if there are similar rings, so this is all idealized, but if you inhibit in a circular area around each one, then the most stable pattern is the one where they are as far as away from each other as possible. Yeah?
Probably you answered, but so Turing patterns are, we have many Turing patterns, right? Depending on boundary conditions and whatever. Yeah. So why, I mean...
Well, there aren't so many. I mean, I mean, most patterns are unstable. So you can get this, or you can get stripes, for example. But this is many places in nature. Then I guess the idea is that it ends up in a hexagonal pattern because, well, again, depending on the boundary conditions, this is the most stable pattern. Yeah.
Are the time courses between excitation and inhibition known? So is inhibition longer lasting than excitation?
Yeah, I mean, these models, time is one of the things that is actually more or less factored out of this. So this is a kind of stable pattern where you assume there is inhibition. We know there are many, many types of inhibition, slow, fast, everything. So I mean, in these models, you don't even consider that. It's very relevant, but I mean, this is extremely simplified. Yeah. All right. But then let me also say that you don't actually need that excitation. You can even do the same with just inhibition. So this is model work from both from Joran Burak and Ila Fiete, and also Yasu Rudi in our institute has worked on that. So you can actually conceptually easily understand that, because if you remove the excitation, all that matters is that you have a ring of inhibition, and again you will then end up with such a pattern. And in that case, it's not a Mexican hat, then you can refer to it as a inverted Lincoln hat. So this just shows that you can actually get such a pattern by simulation, so I don't want to spend time on that. But again, the point is that even here you can do the same, namely you introduce information about the animal's speed and direction, and then you can actually move the bumps of activity around in the network in accordance with the animal's position in the environment. So this is a very, very simplified, short introduction to these models, but it is still, things have changed actually. I mean, in the beginning, after grid cells were found, then actually 2-3 models came up immediately. So this is one. Another one, which you may or may not even have heard about yesterday. Did you skip them, or? The oscillatory interference model. Yeah, so they were very popular for some years, but I think, for example, Nahum's work has pretty much ruled them out. There are also many other things that don't fit, so I won't spend any time on it. And there are yet other models that use completely different mechanisms. For example, again, I don't know if you mentioned that, but adaptation is one mechanism. So if you have cells that show strong adaptation, and at the same time you have Hebbian plasticity in the network, there's also one model that suggests you can get grid cells. But in order to get some of the properties of grid cells, you still need to introduce those recurrent connections in the end. So some sort of attractor mechanism probably has to be there. But then what are the predictions? Is there any evidence that it might be like this? Because this is still very far from the data. There are at least 2-3 observations that I wanted to mention. So one is if you have such a network that I can just go back now to this. If you, or let's say here, if you want to move in, if you want to have correspondence between the movement in the outer world and the movement in the network, then it would be very, very complicated for this to. It would be very complicated if there wasn't some correspondence between how far the animal moves or what direction it moves and how far it actually moves in the network. And that means that if there is a graded scale of grid cells, if there are grid cells of different scales within the same attractor network, it gets quite complicated to get that work, or almost impossible. So therefore there has been a suggestion that for these attractor models to work, the cells within the network must have similar spacing and they must have a similar orientation. Then they work, otherwise they won't work. So this was suggested almost immediately. It was kind of implicit in the models. But then if the cells in the network have the same spacing and the same orientation, that means that it must be a modular organization. You can't have a slowly changing scale of grid cells. There has to be groups of attractor networks. And that was then confirmed with this slide that I believe Nahum showed yesterday, which then shows that the size or scale of grid cells, that means the distance between the peaks, plotted as a function of, in this case, how far you go from dorsal to ventral, then you find that the scale increases, so each dot is one cell, but you see that it is in a step-like manner. And then within each of these modules, not only have a similar spacing, but the cells also have a similar orientation, as would be predicted by these types of models. The spatial relationship between them, that's another story. I won't go into that, but there is some consistency in that too. But I think the idea about the attractors, the prediction was primarily that it has to be organized in modules. And then the other prediction that was has been made goes back to what I showed for the ring attractors, namely if there is such a ring attractor to explain the head direction cells, then one of the observations you would predict is that cells that have a similar orientation in one environment will also have a similar orientation in another environment. In other words, that the network is coherent across environments. So is this the case for grid cells? Well, it seems to be. So I'll try to explain this. This is a figure from a long time ago, from 2007, some of the early work we did. What Marianne Fien and the others who participated in this study did was to train rats in two environments. It was a square environment and was a circular environment. And then she recorded lots of grid cells in both of them, and then compared the grid cells in each of them. And then asking if is there any consistent spatial relationship between the cells pattern in this environment and in that environment. So in order to measure that, she used cross correlations, something we did in collaboration with Alessandro Treves, and generated cross correlation plots for the grids here for both two repetitions of the same environment, two different trials in the square box, or tests in the square versus tests in the circle. And of course, it's the latter that is interesting. But let's first look at the two repeated trials in the same environment. So these are for different cells, 7 different cells. And what you see here is that you get a grid pattern. The cross correlation is a grid pattern with a peak just at the center. And that just means that the grid actually is in the same place for each of the cells. That's not surprising. It just means the grid is stable. But if you now compare the grid in the square and in the circle, you also see still a grid pattern, but the peak is now is not exactly at the center, but it displaced, in this case, slightly to the right, maybe 10-15 centimeters, in this cell, in that cell, in that cell, in that cell, and that, and that. So all of these cells actually are displaced similarly in the same direction, same distance, from this environment to that environment. So it's a shift of the grid from the A environment to the B environment that is exactly similar in all of these cells that were recorded. So that suggests that there is the grid map remains one coherent map that just makes one similar change from one environment to the other. At that time, we didn't know about modules, and that has been shown later, but luckily, in a sense, here were almost all cells from the same module, so that they all shifted similarly, they all behaved in the same way. And that could even be seen in population cross-correlations. You just stack many cells on top of each other and correlate them using population vectors. And again, not only here when you compare A and A, but also when you compare A and B, you get very nice grid patterns, which means that even if you average across so many cells, you still have a nice grid pattern, a consistent shift, which then again suggests that the whole population is shifted from one environment to the other. So that's what people often refer to when they say that the grid map is low dimensional. That just means that it has one configuration that is used over and over between different environments. And this is very, very different from place cells in hippocampus, because that's shown here. So what, again, similar approach, population vectors, you just stack the maps for many cells on top of each other. So what we did here is to do that for all the simultaneously recorded place cells. So now it's just one peak because it's place cells. And then correlate place cell maps from 11 different rooms. So that's what on the axis in this matrix. So the symbols here mean familiar room 1. There was one familiar room, and then novel room 1, 2, 3, 4, 5, and then again 1 second time, and then familiar room 2, and then novel 6, 7, 8, 9, 10, and N6. And same here, and then color for each combination. There's a color code, and the color refers to the correlation between the stack of maps in the two rooms. And what you easily see here is that everything is essentially blue, shades of blue. That means that's a very, very low correlation between place cells in any combination of rooms. However, there are a few exceptions, and of course disregard the diagonal because correlation with itself gives maximum correlation. But there are a few exceptions, those that are marked with asterisks here, because those, maybe you see it, are the ones where you actually correlate the same room, just different trials in the same room. So that applies here, here N1, N1, and here N1, N1. But otherwise, everything is blue, which means that in the hippocampus, in place cells, the combination of active cells in any given room, or in any given environment, is as different as it can be. It's not different from what you would get by chance. Whereas in the entorhinal cortex, then, it's completely different because you retain the same pattern of activity between cells from one environment to the other. So this map in the entorhinal cortex is low dimensional. I mean, it keeps the same correlation structure. And this map with the place cells is, in a sense, high dimensional because you have so many configurations of it. And I think then going back to the question about the place cells, I think that is a major feature of hippocampal cells that's actually more important than the fact that it responds to place. Yeah. But then, with that in mind, then we ask how general is this low dimensionality? So does it extend across behavioral states, for example? So Richard Gardner in our lab then tried to approach this by recording from grid cells in several states, not only when they run around in a box, but also during two sleep states, slow wave sleep and REM sleep. And the question was then, I mean, of course he can't measure grid patterns when rats are sleeping in one position, but what he can do is to measure the cross-correlation pattern, the temporal cross-correlation between cells. So in other words, you can ask, if two grid cells are active in the same places, having the spots in exactly the same place, And that means that they're always coactive, and we know that would happen in both one environment and in another environment. Then you can ask, does this also happen in sleep? So those cells that are active together in the awake state, maybe they even retain their cross-correlation pattern when the rat is sleeping. So he then distinguished between cells that had different degrees of correlation. So cells that were in phase, shown here. So these are 2 grid cells recorded in a box here. And you can see that the grid patterns are almost identical, 2 different cells. They have the peaks in the same places. And then, of course, if you correlate them over time, make a temporal cross-correlation between those 2 cells, they have a big peak here at 0. So it just means that if one cell fires, then it's very likely that the other cell fires too. So much higher than at any other given time. So you have the peak here. That's just as expected. But if you now go to sleep, this is what you see. So you see a similar peak here as well. That means that coactive cells here are also coactive in sleep. And similarly, or conversely, if they're out of phase, like this cell, and then of course you expect when they're running around in a box that there is a dip here. I mean, one being active means that it's unlikely that the other is active. Here, for example, there. And this is again retained in sleep. So if you do that for the whole population with all possible combinations of pairs of cells, and then organize them first in the awake environment from those that are most highly correlated, and then color shows the correlation, and then each cell is 1 row, each cell pair is 1 row, then you go down and you get more and more and more or less and less and less correlation. So this is just how you organize the awake data. But if you then go to the sleep state, you see actually that it is largely retained. At least those that are most correlated here are in the awake state are also most correlated in the sleep state. And same thing also in REM sleep, so that it seems like even in sleep, in all possible states, 2 seconds, in all possible states, then it is one correlation structure that seems to be expressed regardless of the state or behavior or condition. Yeah, now there were several. You first and then you.
How would you interpret the missing negative correlation in slow sleep?
You see, this you mean? Yeah, well, the fact that it's less, you mean? Yeah. I think, well, I don't know, we haven't looked directly into it, but I think these almost all the activity here is taken from sharp wave ripples, so the whole population really is excited. So it could be related to the fact that then you have a general excitation of the network. So, but it still is maybe not so clear, but you still have inverse correlations here. It's just weaker.
I'm wondering to which values they are normalized, because your scale goes up to 2.3.
Which way they are all normalized? That I don't know, actually. Then we have to ask Richard. Yeah, they are normalized, but why 2.3, that I can't tell you. Yeah.
Probably to the mean. Then you could do something like that. Probably normalized to the mean.
I would assume so, yeah. Yeah. Good, yeah. You and then you. Yeah.
You have a really different time scales on the open.
Yeah, good point. Yeah, I forgot to say. So the time scales are very different. Here you have the examples. Yeah, so during slow wave sleep, actually it is accelerated pretty much. So, but still, those cells that are correlated highly in the awake state are still correlated, but the time scale is, things are going much, much faster. Yeah.
My question is a bit more general. So about the distribution of the different phases of the grid cells. So for like one scale population. So how, I mean, so you're saying that they were just randomly labeled, or more than, like, they were not all together, right? But are they actually not random, but explicitly such that they are not close to each other?
Yeah, you mean whether in the wake state?
Yeah, I guess because it doesn't matter, but like,
Yeah.
they
No,
basically are two anatomically close cells, or maybe, I don't know if you know the connectivity, because that would be more interesting. Similar cells, are they more than randomly expected different from each other in the face?
Yeah, I mean, we have to some extent looked into this. So I mean, the two questions. First, are all faces equally represented, or is there some sort of clustering regardless of anatomical location? It seems like there is no clustering. We thought that for a while, but then as we got more data, it actually seems to be pretty distributed. But then regarding anatomical location, so by and large, it is pretty random. But if you go really, really close, then there are data from studies using calcium imaging, where you can then know the anatomical location of the cells. This is data particularly from, there's a study from Dombeck, where you actually, and we have seen the same in our own data, that cells that are very close are more similar in phase. But that means very close means less than 50 microns, so it's just 1 or 2 cells away. So there's some patterning, but it's quite weak. So that means that for these attractor networks to function, it has to be, the connectivity is what matters, so that cells with similar phases have to be connected. That's the only way you can, like in a ring attractor, cells that have similar orientations are connected, then grid cells that have similar phases should have stronger connections. There is some indirect evidence for this, but using quite sophisticated statistical methods. But of course, this is something that we are trying to observe with much more direct methods. So I think the anatomical location of the cells isn't really what matters. What matters is whether cells that are have a similar grid phase are actually connected. That's an explicit prediction of those networks, and I mean, hopefully we'll find out in the next years whether that is the case. But it's a clear prediction for this to work. So now let me have to coordinate time. How much left? 40? 45?
Yeah, so we started at,
Okay, so let me spend some time on this, and then I'll spend more time on the object vector cells, and then we'll see. So as I'm sure you heard from Nahum yesterday, then these data are from very, very simplified environments, and that is both good. The good thing is that otherwise we will never have detected the cells. Yeah, but the bad thing is that it's not. I mean, the real world is slightly more complicated. So, an obvious question is to ask how do grid cells and also the other types of cells actually operate in more normal environments. So, one of the questions you may ask is, for example, and which was asked early on, is there a continuous grid? I mean, do grid cells have one grid pattern that just extends from here to Siberia? That's not the case because environments are compartmentalized like houses. They consist of rooms that are pretty much individual units. So one way we did address that for many years ago together with Dori Derdikman was that he trained rats first in an open field, just like you have seen, but then put them into a maze, which we call the Hairpin Mazes. They go in a zigzag pattern from one end to the other, and then they went back. And the basic result is the following. First, in the open environments, you get a normal grid pattern. So that's what you see at the ends. But the more interesting thing is what you see in the middle, in the zigzag or the hairpin maze, because you don't get an overall grid pattern here anymore. What he found was that each time the rat made a turn and went down an alley, then a certain pattern unfolded, still periodic peaks, more or less periodic, but the same pattern seemed to repeat itself for each alley as long as the rat went in the same direction. You can see it's particularly clearly here, maybe. The rat goes up here and then walks a certain distance down. You get peak of activity and then goes down, gets up again, and the cell fires here, actually in this case in both directions in the same place. But you can see that for each lap, for each alley, it's the same pattern that unfolds. So you don't see a grid pattern on top of this anymore. So what it suggested was that the grid pattern actually is kind of reset each time the rat makes a turn and goes into a new sub-environment. So that led to the idea that these grid patterns are actually fragmented, and that in the real world you have local grid patterns that are somehow merged together. That's also suggested by data from larger environments in 2 dimensions. So, and including the cell I showed early on, that if you watch closely, you can actually see that the grid pattern isn't as regular as you might think. So for example, you can see that is a fairly regular pattern in one part of the environment and in another part of the environment, but then the orientations, for example, might be different, and it seems like the pattern is kind of broken in the middle in this case. And this is, of course, because of distortions or influences from the walls, which you heard about. I won't really go into that, but I want to use that as a background for asking how can, if grid patterns actually consist of small periodic areas that are somehow fused together, how can this be fused together? So Tanja Werner in our lab, whom I showed early on here, she trained rats in 2 boxes, 2 rectangular boxes, and then she fused the boxes by removing the central division here after a while, and then asked what happens. Do you retain the original grid patterns, or is one grid pattern, is it a winner-take-all effect, where one is just extended into the other? Or is there some sort of integration of the patterns into a single grid, or is a completely new map formed? So many possibilities. So what she did was that she compared then grid cells recorded in the rectangular spaces with the big open space. And then she used the sliding correlation procedure, beginning with a small window and just moving and comparing for each window the locations in the rectangular boxes with the corresponding locations in the open box, and then sliding this window from across every possible position in the environment. And then what she found was that is then color-coded, so that's a correlation. Red is high correlation, blue is low correlation. And this shows the result for a single cell. So high correlations, especially at the top and at the bottom, but lower correlations in the middle. And this becomes clearer as you average the cells, like you see here. And if you average all of the cells from all of the rats, you get a clear band here in the middle, which shows that there is a lot of change in the middle here, where you remove the wall, whereas the cells actually keep their grid patterns at the upper and lower ends here. That's also shown here. So what kind of reorganization takes place in the middle there? Well, she investigated that in 2 ways. So first, by identifying the fields using a local field detection procedure. And then, so what she did was she identified the fields both in the rectangles and in the open environment. And then she asked for each field in, let's say, in the rectangles. She then asked where is the nearest neighbor within a certain circle. And then she defined vectors for each of them to the nearest neighbors. And then plotted all these vectors and found that the vectors are longest in the center, then again suggesting that fields move or are displaced more in the center and not much in the periphery. And she did the same using a different procedure. I won't spend time on that, but you can also use a cross-correlation procedure and again find the same, most shift or most reorganization in the center. And then the question comes up, what kind of reorganization then takes place actually in the middle there? Could it be that what happens is that you actually locally obtain a new grid where the wall previously was present? So what you did was to ask what is the distance to each of the nearest neighbors field, even again within a certain predefined circular area. And then she measured the variation or the standard deviation between those distances. And then the standard for each sub-area of the environment. And then you can see again that in the center, then before wall removal, not surprisingly, there's a high standard deviation because the two maps on the two sides are really split. So you may sometimes have really clear breaks in the grid pattern here. But afterwards, this disappears. So that means that you have low standard deviation. That means that the grid distances are very similar, meaning again that you have you're getting a grid pattern here, and that any deviations are actually rather along the walls, as you would expect if the walls have strong influences. So that just suggests then that it's consistent with the idea that grid patterns in larger environments or more complex environments consist of local grids that then are fused together and become locally coherent, but then as you go over longer distances, then the coherence is no longer there. But still, it is probably coherent over a sufficient space that you will never have points where you, if the rat uses path integration to determine its position, where you suddenly have a big jump. So there's always locally at least some coherence between the actual movement of the rat and the change in the grid pattern. And that may relate back to what you actually heard also in the previous talk, that but also the rats use correct in this case their grid pattern, or correct the path integrator by using visual information constantly, so they always refresh. And if that is the case, maybe you don't need a grid map that goes from here to Siberia, because you only need it for a little while, because under normal conditions, very soon, then you have the opportunity to correct the position of the grid map, not only by vision, but actually, I mean, as you all know, rats are good, can use odor cues, they can use auditory cues, anything.
What does produce this effect of this integration of the grid map cells? I mean, is the direct experience of the rat moving in the environment, or are visual cues? I mean, the partition is opaque, or it happens also with the transparent partition?
Oh yeah. No, we actually tried a transparent partition at some point. We don't have much data, so I can't say. But I mean, based on the evidence we have from various types of environment, then it really matters that the rats can actually walk over there, because it uses, it's a major factor that the rat probably uses to create coherence of the grid over a larger distance, is its own self-motion. But I mean, I still think it is, well, I think you can probably, with some experience, actually infer locations. That's related to what you're doing, David, isn't it? But I think, by and large, the most important one is movement. So it certainly helps that they can actually walk from one space to the other. Good. Yeah, I'll skip that. This is just to show that the two maps remain coherent. But I think I will make sure I have enough time for the next section. So with that, then I'll skip the last 2/3 of my talk. No, not quite. 52. I made it to the number 52 out of 70. But I had planned to tell you about interneural networks. No time for that. And maybe, I think very exciting, is the encoding of time in the entorhinal cortex. But that's time, and the topic of this meeting is space, so maybe it's okay that I skip it. Good. Many people, I think you can read them. I mentioned the key persons on each of the projects as I went through it. So with that, I think now I'm 1 minute ahead. Well, that's it. Thank you.